Hello, everyone. Welcome to this webinar, and thank you for all your time. My name is Angel, and I'm a product manager from Crystal Bio. It's my pleasure here and to have this chance to meet you in this webinar and share knowledge about recombinant protein DT3C and its role in analyzing the antibody internalization efficiency in ADC drug development. After my presentation, please feel free to ask questions and share ideas with us. So go back to topic today. So today we are going to talk about the recombinant protein DT3C, and to introduce that is as a convenient tool for the antibody internalization analysis. For today's talk, I will briefly introduce the development of ADC and some current ADC product, and also to emphasize the analysis tools that related to the antibody internalization. And lastly, I will introduce our protein, the recombinant DT3C, and how it's applied in the ADC drug discovery. So, as a new class of anti-tumor medicine, you may know that the ADC drug is developed a lot. So it provides a means to reduce the off-tumor toxicity by limiting the payload exposure in normal tissues. And here I put a very prediction about the global sales of some selected approved ADC drugs from 2020 to 2006. And as you see here, this blue line representing the drug from the AstraZeneca and the Daiichi Sankyo is called the TDXD. The target of this one is targeting the antigen HER2. As you see, it has a predicted sales of more than 6 million by 2026. And the other one, the pink one here, is representing a drug targeting the Nectin-4, and it's called the Padcev. So as you see, the increasement is also large. According to some new analysis from Data and Intelligence, the global ADC market size was valued at $5 billion in 2012 and predict to be more by 2030. So looks like the growth rate is huge for the ADC market. And actually, it's not only for the current ADC drug, but also for the clinical landscape. According to the summary I put here, looks like that future ADC is still being hot. From 1997, look like over 1,200 clinical trials have been registered to evaluate the safety and efficacy of different ADCs. So I really hope that with this clinical trial of ADC, the future ADC will bring more benefit to all the cancer patients. So as I talk about the ADC, you may be wondering that what are ADC and how do they work? So ADC is very hot in the oncology market. And actually it's very complicated. Here I put a very traditional structure about the ADC. ADC have 3 key components that you may need to remember. The first one is this big molecule here is monoclonal antibody. And here is the stable chemical linker. At the end of the linker, you will see the payload, which carrying the cytotoxic drugs. So a monoclonal antibody will bind to the antigen at the surface. And this antigen is preferentially expressed on the tumor cell surface to make sure that this molecular antibody will specifically binding to the tumor cells. And also with the covalent linker here, it can make sure that the whole complex is stable when it's circulating in the bloodstream. And also, the payload is the important key component that will induce the tumor cells to be death. So looks like that binding of the molecular antibody to the target antigen results the whole ADC complex enter into the cells. And actually, it is. So here is a very brief description about the mechanism of the action. So I will describe the steps briefly. So the whole ADC complex, because the monoclonal antibody will bind to the antigen, so the ADC complex can enter into the cells. So inside the cells, the antibody portion will be degraded inside the cells. And after degradation, the free payload will be released inside the cytoplasm. And a fraction of this payload will interact with the target, resulting in the cell death. And you see that partial of the free payload can be taken up by the neighboring cells. And this whole process was known as the bystander effect. So with the bystander effect, some cells surrounding the target cancer cells can also be killed independently of the target antigen expression. So it looks like that the ADC have a more benefit in the cancer treatment. So the success of ADC in inducing the cytotoxic depends on various factors. The three key components that I mentioned here, they are definitely very important, but there are still some other factors that may affect the safety and efficacy of ADC, such as the linker development and also the conjugation method between the linker and the payload. All this, they are kind of like lead to the success of ADC. To have a better understanding of the ADC, here I showed some approved ADC here from two different categories. So the top 6, they are for the hematological tumors, and the bottom 7, they are for the solid tumors. I'm not going to talk all 15 current ADC drugs in the market, but I want to use this figure here to highlight some features about ADC. So the first ADC drug I want to talk about is the one that I mentioned before, which has a very sharp increase in the prediction. This one is the T-DXd. So for the T-DXd, in my view, the main feature is that the ratio. The DAR ratio is 8. So the number here representing the DAR ratio. So you may wondering what is DAR? DAR, the full name is the ratio between the drug and the antibody. So it means that the antibody how many payload molecules that this antibody can carry. So the number 8 here means that for this drug, each antibody can carry 6 DXd, and the DXd is the payload. So from the big number here comparing to the others, you kind of can imagine that the efficacy should be pretty good, and actually it is. And another drug I want to mention is this one, so the Nectin-4. This one is called the PADCEV. So for the PADCEV, the main feature that I want to highlight is that the Nectin-4, which I said, the Nectin-4 is the target. So comparing to the T-DXd, this one is targeting a different antigen. So it looks like that in the ADC market, people are expanding the choice of the antigen to kind of like discover some novel therapies here. And actually it's very, very good news. So overall, from the current approved ADC drug, we see the similarity and the difference. We are glad to see the expanding of the antigen choice, and we are also happy to see the advanced development of the linker technology and the payload novelty. All this, they kind of like lead to the next generation of the ADC development. And we are also sure that it's definitely the next generation of ADC will be better. So the ADC development is dramatic. However, for the next generation of ADC, the challenge still exists. So according to this mechanism of action of internalizing ADC here, after the ADC is internalized, we can see that only after the internalization, the complex will be degraded, and only after the degradation, the payload can be fully released. So it looks like that there's a correlation or direct influence between the internalization rate and the efficient payload released. And actually, it's true. Early studies have been showed that there's an intricate correlation between the antibody and the tissue penetration and as well as the protein binding affinity. So most current ADC drugs, they are designed against a target antigen that display efficient internalization to facilitate the ADC entry upon recognition. So the challenge that I will talk in this slide is actually related to the antibody internalization efficiency. It's really important about this internalization efficiency because that for different antibodies, their efficiency can be different. So which means that you probably need to assess the internalization efficiency if you want to choose an optimal antibody, and also choose a good pair between the antibody and antigen will make sure that your internalization is kind of like ADC compatible. And inefficient internalization actually is not that good because it will lead to off-target release of ADC and causing some toxicity to the body. And not only the toxicity, but also the efficacy. So early release of the payload also resulting in the decrease of the efficacy. So from this view, you kind of can relate, you can kind of think that the internalization efficiency is very important during the development of ADC. So with this thought, various assessment tools have been developed to facilitate the ADC discovery. Here I list the different conventional methods that they are used to detect antibody internalization. I briefly categorize them into three different systems. So like the classic antibody screening containing the pH-based and the temperature shift-based. And the second is the live cell imaging-based antibody analysis, which can kind of like real-time monitor the location of the naked antibody. And the last system is based on the toxin conjugated killing detection and analysis. So I will talk one by one in my next part to introduce this different assessment tool. So the first one is the pH-based from the classic antibody screening. So this one is pretty traditional method. And for this method, the basic idea is to use a hydrophilic and bright pH sensor dye. And this pH sensor dye is kind of like not fluorescent at the neutral pH, but it will become highly fluorescent when it enters into the cells, because inside the cells, the environment there is kind of like acidic. So because the pH change, the pH sensor dye will change from non-fluorescent to fluorescent. And usually the pH sensor dye is conjugated to the Fab fragments, and this conjugated Fab fragments is linked to the primary antibody. So when the primary antibody entering into the surface, because the pH change, so the attached sensor dye will cause the intensity change during the whole process. So with this, the researchers can kind of like determine like what is the extent of the internalization. And some output is here. So here I put a figure showing that how the fluorescent intensity will change. So briefly, during the neutral pH, the fluorescent signal is low, but after it enters into the cells due to the pH change, the acid condition, the dye will become more fluorescent. For clarity, this figure showing here is not from a real data. I just want to show what the trends that you're expecting to see. And the next one is the temperature shift-based screening assay. This one is kind of like similar to the pH, but instead of the pH change, you kind of like see the intensity signal change based on the temperature. So during the 4 degree, the antibody is binding to the surface, and after it changed to the 37 degree, the surface antibody can be internalized into the cells. Then after the surface quenching, the phenotyping antibody will become frozen. Then you kind of like can imagine that inside the cell and outside the cell, the signal of the fluorescent is different. So based on the intensity change outside and inside, the researchers can distinguish the fluorescent signals and to determine that how the antibody are internalized and then what is the efficiency of the internalized. So it's kind of like similar to the pH. However, it's not that popular as the pH one. So the next one is and actually the live cell antibody internalization analysis is kind of like based on the pH screening assay. So for this advanced analysis, it's kind of like a time and concentration dependent internalization assessment tool, because with this one, you can use with different equipment, such as the confocal microscope I put here. But instead of using the microscope, you can also use the imaging-based system or some other compatible instrument. So with this, the researchers can do a monitor about the real-time observation. So with this, you can see that how the antibody the whole process of the antibody entering from the outside of the cells, entering into the inside of the cells. So it's kind of like a really exciting to see the localization of the antibodies. And for the whole process, as the time goes on, you can kind of like see the ratio or the extent of the internalization of these naked antibodies. And for the last one, the toxin-based screening here. So this one is use the immunotoxin to cause the toxicity to the cells. And by causing the cell death, you can refer back what is the kind of like an internalization status of internalization efficiency of the antibodies. It relies on a release of the toxin inside the cells. So here's one example. So you attach the toxin to the monoclonal antibody, and because the antibody is binding to the surface antigen, then the whole immunotoxin is internalized into the cells. And because the furin inside the cytoplasma, the furin will cause or inactivate the EF2, causing the inhibition of the protein translation, then causing the cell death. So when you hear this, doesn't that sound familiar? Yes, you are right. So the mechanism is kind of like similar to the ADC. So instead of using the payload in the ADC here, this assay use a toxin to replace the payload. So this assay kind of like can be processed in a high throughput manner, and is usually commonly used for some initial screening. So before I move on to the next part, here I want to do a simple comparison between all three assessment tools that I mentioned. So for different analysis types, they may have different features, and also they have different applications. For example, like the classic one, the feature about this type of assay is that it's high specific, and also the experiment can be quantified. You can finish the experiment with the flow cytometry as well as the fluorescent microscope. And for the live cell checking analysis, because it's a real-time observation, so it can do some time-dependent analysis. But with this one, if you need to finish the assay, you probably need some additional system, like the imaging-based system or some compatible instrument. So it may be a very, like, increase the cost when you finish the experiment. But for the last one, it's kind of like more economic than the previous two, because for this one, if you want to do a primary and initial screening about the ADC compatible antibody, you can do that with this one in an economic way. And because this one can be performed in a high throughput manner, so it's kind of like it can decrease your cost comparing to the live cell. And also the application for this one is that you can do the assessment just with simple cytotoxicity assays to get a kind of like an initial prediction about the antibody internalization efficiency. So based on this, I will stay at the immunotoxin-based screening to introduce more details and more correlated applications and how they are used in the ADC discovery. So for this type of the screening assay about the antibody, so basically there are two different products. One is the MapZap, and the other one is the recombinant protein DT3C from our Crystal Bio company. So for the MapZap, as I showed here, so the MapZap have three different parts. So first, it needs to form a second immunotoxin with the secondary antibody and the set pairing. So the second immunotoxin can bind to the primary antibody that you want to screen because of the second antibody. So basically the idea is that second antibody is binding to the primary antibody. And during the pole binding process, the set pairing is also carried. Because it's covalent linked with the second antibody. So all three can be combined together. But comparing to the MapZap, recombinant protein DT3C is more convenient and actually is more simple because itself is already carrying all components that you need to, because for the DT3C, actually the toxin part is a differing toxin without a receptor binding domain. And instead of using the original receptor binding domain, the 3C domain of the Streptococcus protein G replaced it. So you can just use this simple recombinant DT3C to bind to the primary antibody to form the immunotoxin. So comparing to this one, you kind of can imagine without the second antibody, the immunotoxin formed here actually has a very smaller size. So smaller size means that during the ADC assessment, the penetration of this immunotoxin will be much better. So here is a very brief illustration about how the DT3C entering into the tumor cells. So as I probably mentioned before, the DT3C kind of like replace the position of the payload, conjugated with the antibody. And when the whole immunotoxin was internalized into the cells, the tumor cells can be caused to be apoptosis. So with the number of the cell deaths, you can kind of like refer back what is the internalization efficiency of this antibody. So with this one, you do not need to make a selection of the secondary antibody conjugates. So you don't need to worry that if your primary antibody is mice, then do I need to use a mice compatible secondary antibody? So no worry about species. It's quite common to multiple species with this recombinant protein DT3C. So it's more economic. And our Crystal Bio already confirmed that the purity of this recombinant protein is very high. So based on our SDS phage and HPLC result, we showed that the purity of this recombinant protein is higher than 90%. To make sure that you can have a better understanding about how this DT3C work, here I put a very general overview about the workflow, like how you use the DT3C to assess the internalization efficiency. Pretty simple. You prepare the antibody and you prepare the DT3C, briefly mix the antibody and the DT3C together. And after 30-minute incubation between antibody and the DT3C, so the temperature of the incubation can be room temperature, also can be 37 degrees. So choose either one. After 30-minute incubation, you will get the immunotoxin formed during the whole incubation. Then you can prepare the dilutions. And the next step is that prepare the cells that you want to test and then place them in the 96-well plate and add antibody and the DT3C conjugates. After another second round of the incubation, you can just simply acquire the data with the cell cytotoxicity assays. So here, for the assay, I put the MTT here, but definitely MTT is not the only choice. You can use some other familiar cytotoxic assays that you used in your labs. So here, the whole experiment is very easy to prepare and is very convenient. DT3C acts as a very common tool to perform the antibody screening in a very high throughput manner. So not only in a very common scenario, here I put another scenario that how the DT3C can facilitate the screening of the functional antibodies that compatible with the ADC property. So the whole idea about DT3C is to facilitate the ADC discovery. So we want to highlight the wider application of this one with the hybridomal technology. So you may be very familiar with this screening pipeline with the hybridomal technology. Basically, you do the immunization with the mice. Then you have the hybridomal library constructed. After the several rounds of the screening process, you perform different dilutions, then you will get a very excellent functional antibody. But with the DT3C, you can even do more. You are not only get a functional antibody, but you also get the functional antibodies compatible with the ADC properties. So here is a kind of example showing that how you do it. So after the hybridomal library constructed, when you do the selection, just incubate the supernatant from the hybridomal library with the DT3C. After all the incubation, you can kind of like to obtain the antibodies that with the excellent internalization capacities. So the final product will be more than the normal antibodies. So during the whole process, it looked like just add one more step, but actually you are saving the time that you may spend in the later ADC development stage. So we put all the discovery in advance at the very beginning to save your time and the money. So it's kind of like a pretty economic to use the DT3C. So finally, I want to highlight the DT3C, especially the advantage of why we need to use it. So DT3C can evaluate the internalization efficiency of antibody in the vitro. And as I said a lot, the preparation of the immunotoxin is quite simple. You mix the monoclonal antibody with the DT3C, simple incubation, just 30 minutes. At room temperature, 37 degrees. Then you can move on to the next step to finish your experiment. And also, comparing to the MapZap, you don't need to worry about the species of your second conjugates because the DT3C can form the immunotoxin with the molecular antibodies from multiple sources. So just one common tool, and then you will finish all. And in the right panel here, it's kind of like an experiment showing that with the DT3C, the cell death is pretty dramatic. So you add the DT3C form the immunotoxin with the molecular antibody. And then after that, the cell death happens. And you can kind of like go from the cell death to determine that what is the efficiency of the internalization. And also comparing to the MapZap, it's not only that the size is smaller, the penetration is better, but also the internalization efficiency is higher that you can get. And here's a kind of like a result that I got from the literature experiment. It showed that with the DT3C, the internalization efficiency is much higher. So lastly, one more take-home message is that DT3C is kind of like a simple, efficient, and economic tool to evaluate the efficiency of antibody internalization, especially when you need to do some primary and initial screening of antibodies. DT3C will be always your best tool to choose. So last part, please give me a few minutes to introduce our company. Our company is Akuso Biotechnology. It was established in December 2007. It's a biological high-tech enterprise integrating the scientific research, product, and the sales. So we have many different reagent and service provided. And here is a layout of the product line that we have. We have the ELISA kits, we have the protein, we have the antibody, and we also provide the molecular biology products and the small molecular antigens. So it looks like that we have a diversity product and service for you to choose. And also our products have already been exported to more than 80 countries, including the Europe, American, Japan, and Germany. We have different cooperations with some famous companies like Pfizer and the Bayer and also the Moderna and AstraZeneca. So it looks like that we are so proud to have this coordination. And thank you so much. And the last thing I want to emphasize is that if you want to apply for a free trial of the DT3C, please scan the QR code to get started. So let the DT3C to facilitate your ADC discovery, and it will be a wonderful journey. Thank you.